Welcome to our journey into the microscopic world of animal diversity. Today, we are going to explore the general characters of Protozoa. If you have never studied these fascinating creatures before, do not worry. We will build our understanding from the ground up. By the end of this session, you will see why these invisible organisms are some of the most important and complex survivors on our planet. Let us dive right in. To start, I want you to imagine a bustling city where millions of people work together to keep things running smoothly. That is very much like a human body, which is made of trillions of specialized cells working in harmony. Now, imagine a single person living alone on an isolated island who has to be the builder, the chef, the doctor, and the waste manager all at once. This incredible one-person army is exactly what a protozoan is in the biological world. They are microscopic, single-celled organisms that perform every single function of life entirely within one tiny cell. They are the ultimate survivalists. Now that we understand their independent nature, let us look at how science formally defines them. Scientifically, protozoa are defined as unicellular, eukaryotic, and primarily heterotrophic organisms that belong to the kingdom Protista. The word itself comes from Greek, where proto means first and zoa means animal. This reflects the historical belief that they were the very first animal-like creatures to evolve. Being eukaryotic is a crucial detail. It means their single cell contains a true membrane-bound nucleus that safely houses their genetic material, unlike simpler bacteria. Because they are heterotrophic, they cannot make their own food through photosynthesis. Instead, they must hunt or scavenge. Knowing what they are naturally leads us to ask why they matter to us. You might wonder why we should care about invisible creatures. The truth is, protozoa are absolutely critical to the survival of our planet. In aquatic ecosystems, they form the foundation of the food web. They eat bacteria and are in turn eaten by larger creatures, eventually feeding the fish that humans consume. Without them, our oceans would become stagnant. On the darker side, many protozoa are dangerous parasites, responsible for devastating human diseases, such as malaria and sleeping sickness. Understanding them is the first step in developing life-saving medicines. Beyond their ecological impact, let us examine how they actually survive day to day, starting with movement. Protozoa have fascinating and diverse ways of moving around their microscopic world. Some, like the famous amoeba, use pseudopodia, which translates to false feet. These are temporary, flowing extensions of their cytoplasm that drag the cell forward. Others, like the paramecium, are covered in thousands of tiny, hair-like structures called cilia that beat in unison, like oars on a boat. A third group uses flagella, which are long, whip-like tails acting like boat motors. These distinct methods allow them to actively hunt for food. Once they move and find food, they need to eat and maintain their internal balance. Most protozoa are holozoic, meaning they ingest solid food particles, like bacteria, whole, digesting them inside specialized internal compartments called food vacuoles. But feeding in water brings a unique challenge. Because many live in freshwater environments, water constantly rushes into their bodies due to a natural process called osmosis. To prevent themselves from swelling up like a balloon and bursting, they possess a vital organelle called a contractile vacuole. This structure acts like a microscopic water pump, constantly collecting excess water and squeezing it back out. Having discussed their internal machinery, let us look at a visual representation of these structures. On your screen is a diagram of an amoeba, a classic example of a protozoan. Notice the irregular shape forming the pseudopodia on the left, which we learned are used for movement. In the center, you can see the large, distinct nucleus that houses the genetic material, making this a true eukaryotic cell. Scattered around are the food vacuoles, where digestion takes place. Most importantly, observe the contractile vacuole, with arrows indicating water being collected before it is pumped out. This single cell truly contains everything it needs to survive. Seeing this complex anatomy brings up an interesting question. Why do they not grow larger? A defining characteristic of all protozoa is that they are microscopic. You might wonder why a single-celled organism cannot grow to the size of a dog. The answer lies in the physics of diffusion. Protozoa do not have lungs or a digestive tract. They rely entirely on molecules diffusing across their outer cell membrane to get oxygen in and waste out. As a cell grows larger, its internal volume increases much faster than its surface area. If it became too large, it would simply suffocate. To truly grasp why they stay small, we must look at the mathematical relationship governing their size. This formula represents the surface area to volume ratio for a perfectly spherical protozoan. The variable r stands for this crucial ratio, and r is the radius of the cell. Conceptually, this formula tells us how much outer membrane is available to service the inside of the cell. Notice that the radius r is in the denominator. This means that as the radius increases, the ratio r decreases dramatically. A high ratio is absolutely necessary for survival, ensuring the membrane can efficiently service the entire inside of the cell. Before we move on, let us see this formula in action with a concrete example. We are given a normal, small spherical protozoan with a radius of 10 micrometers. We want to calculate its surface area to volume ratio to see how efficient it is. We choose our simplified ratio formula and substitute the radius of 10 into the denominator. Dividing 3 by 10 gives us 0.3. This relatively large number means there is plenty of surface area available to service the internal volume. The organism can easily breathe and eat through its membrane without any internal blockages. Now, let us compare that to a hypothetical giant protozoan to see what happens when the cell grows. Here, we are given a radius of 100 micrometers, which is 10 times larger than our previous cell. We use the exact same formula and plug in 100 for the radius. The result drops drastically to 0.03. This is 10 times smaller than the normal cell. Physically, this proves that the giant cell has very little membrane surface area relative to its massive internal volume. It would not be able to absorb oxygen fast enough to keep its center alive. Now that we have used this formula to prove why giant cells cannot exist, let us understand where the formula comes from. We are setting out to derive the simplified ratio formula for a spherical cell. We start with the fundamental definition. The ratio R equals the total surface area S divided by the internal volume V. From basic geometry, we know the surface area of a sphere is 4 pi r squared. The volume is 4/3 pi r cubed. By substituting these geometric formulas into our ratio equation, we set up the mathematical foundation. This chunk connects the biological concept of diffusion to pure geometry. Continuing from our setup, we will now simplify the expression to find our final result. We have 4 pi r squared divided by 4/3 pi r cubed. First, we can cancel out the common factor of 4 pi from both the top and the bottom. Next, we divide out r squared, which leaves a single r in the denominator. This leaves us with 1 over 1/3 r. To clean up this complex fraction, we multiply the top and bottom by 3. This gives us our final elegant result. R equals 3 divided by r. This mathematical reasoning perfectly explains the biological size limit we saw earlier. Having established why they must remain small, let us look at another consequence of their simplicity: how they multiply. Another general character of protozoa is their ability to reproduce rapidly through a process called binary fission. In this process, one parent cell splits equally into 2 identical daughter cells. Because they do not have complex bodies to build, this division happens very quickly. This leads to exponential population growth, where the numbers double with every generation. Understanding this rapid reproduction is crucial because it explains the terrifying speed at which microscopic infections, like malaria, can spread inside a host. Now that we understand they divide rapidly, we use a specific mathematical model to predict this multiplication. This formula calculates the exponential growth of a population undergoing binary fission. Let us break down the symbols. N represents the final population count that we want to find. N0 is the initial population we start with. The variable n represents the number of generations, or how many times the cells have divided. The base is 2 because each cell splits into exactly 2 cells. Physically, as the number of divisions n increases, the final population n grows explosively, doubling every single cycle. Let us apply this growth model to a real scenario in a freshwater pond. We are given that a single paramecium divides once every 8 hours, and we want to observe it over a full 24 hours. Before we can find the final population, we must first determine the number of generations, which is our variable n. We do this by dividing the total elapsed time by the time it takes for one single division. Plugging in 24 divided by 8, we find that the protozoan will undergo exactly 3 complete cycles of division. Now that we have our number of generations, we can find the final count. We start with just one initial cell, so N0 is 1. We just calculated that n equals 3 divisions. We choose our exponential growth formula and substitute these values in. 1 times 2 to the power of 3 gives us 8. While 8 cells might seem small, remember that this rapid exponential growth continues continuously in nature. Within just a few days, this single cell can become millions, illustrating exactly how quickly a protozoan infection can multiply if left untreated. We have covered a lot of ground today, so let us reinforce the most important ideas. First, protozoa are remarkable single-celled eukaryotes that act as independent microscopic animals using specialized organelles to survive. Second, we proved mathematically that their microscopic size is strictly dictated by the necessity of a high surface area to volume ratio for efficient diffusion. Finally, their ability to reproduce exponentially makes them a powerful force, forming the foundation of aquatic ecosystems and driving critical human diseases. You now have a solid foundation in the general characters of protozoa. Great job following along.